This research is carried out in Cabo Verde in West Africa, and the results are important because it gives us an understanding of the levels of microplastic pollution. Today we're going to collect microplastic samples from the Bay of Mindelo, and we've got all our instruments set. We have these microplastic nets. So what we're going to do, we're going to leave these nets in the water, and then tow slowly at a speed of 5 knots and collect the water samples. This research was to assess the levels of microplastics in our water samples and our fish samples, to establish a relationship between these two and also to find out how these plastics affect the fishes.
By this we can collect data about the environmental pollution with microplastics in these oceanic environments, and that's a very important thing to do because plastic litter in general is a very unfair form of environmental pollution because it can be transported with ocean currents long distances and reaches places which are far from the sources of plastic pollution. We are still lacking data, in particular from oceanic archipelagos, and we need to generate baselines.
We have our fish samples. So what we're going to do now is to get the measurement thermophometry. So then we get the guards which we need for the analysis.
Wise is participating in the WASCAL program, and as a WASCAL student he is visiting GEOMAR to do a part of his analytical work for his master thesis here in our laboratories, and I'm supporting him.
The collaboration is very key, and that is what I would say is special to this program.
Analyzing microplastics in environmental samples is challenging, it's technically challenging, and it's much easier if you do this in collaborations.
The results from our research show the high prevalence of microplastics in our fish and our water samples, and if this is to persist, it poses great concerns to the health of our environment, especially the fishes, and the health of consumers of these fishes.